Today we will be going to discuss about ketone bodies. Please listen to this video carefully. This will help you a lot. Basically, we have three types of ketone bodies. It is acetone, acetoacetate, and beta hydroxy butyrate. Okay. Acetone and the acetoacetate basically consist a keto group, and beta hydroxy butyrate don't have any keto group. Okay. Acetone basically have a fruity odor. Okay, I will write down here. Acetone has a fruity odor. First of all, we should know that there are three types of ketone bodies. It is acetone, acetoacetate, and beta hydroxybutyrate. The first two contains the keto group, and the beta hydroxybutyrate doesn't contain any keto group. Acetone produced in the body is useless, and acetoacetate is first to be formed. I will explain how it is first to be formed. Okay, you should remember how that acetoacetate is first to be formed. Okay, so basically the ketone bodies are water soluble. Okay, they are utilized by our brain, and they are the product of metabolism of fats. Basically what happens when there is a breakdown of fatty acid, there will be the production of acetyl coenzyme, and all of you now know that acetyl coenzyme is a 2-carbon compound. Okay, so let's begin with the formation of ketone bodies. What happens? Acetyl coenzyme A combines with acetyl coenzyme A to form acetyl coenzyme A. Okay, now you should remember what is the enzyme involved, that is called beta thiolase. Okay, next acetoacyl coenzyme A combines with another acetyl coenzyme A molecule to form HMG coenzyme A with the help of an enzyme, that is HMG coenzyme A synthase. Okay, then HMG coenzyme A will form acetoacetate with the help of enzyme HMG coenzyme A lyase, and further acetoacetate will form acetone and beta hydroxy butyrate. So that's how first acetoacetate is formed, and further it forms acetone and the beta hydroxy butyrate. You should remember these enzymes involved in the various steps, and most importantly, this HMG coenzyme A synthase is the rate limiting step. Okay, so this was how the acetoacetate was first formed, rather than acetone and the beta hydroxy butyrate. And earlier I have also mentioned that ketone bodies were utilized by the brain. So where they were produced? They were produced in liver. Okay, but liver lacks an enzyme that is called thiolase. Okay, liver lacks an enzyme that is called as thiolase. Okay, and but this enzyme thiolase is present in our brain. In the brain it is present, but in the case of liver it is absent. So the ketone bodies is only utilized by our brain but not by the liver. Okay, so this was the importance of ketone bodies that you should remember. Apart from ketone bodies, in today's video we will discuss about cholesterol. Let me zoom it out. Okay, now we will discuss about cholesterol. If you have understood something about the ketone bodies, it will be easier to remember about the cholesterol. Now let's begin with cholesterol. First of all, cholesterol is a 27 carbon containing compound. Okay, all of the steps are similar, but there are some minute difference that you should get it noted down. Here also acetyl coenzyme A combines with acetyl coenzyme A and form acetoacetyl coenzyme A. Then acetoacetyl coenzyme A will combine with another molecule of acetyl coenzyme A to form HMG coenzyme A. Then HMG coenzyme A here will form mevalonate. This mevalonate will further form isoprenoid units. Isoprenoid units will further form squalene. Squalene will further form lanstirol, and lanstirol will further form cholesterol. If you have remembered the cycle of the formation of ketone bodies, then it will be simpler to remember about the cholesterol. So basically, the two molecules of acetoacetyl coenzyme A form acetoacetyl coenzyme A with the help of enzyme beta thiolase. Then acetoacetyl coenzyme A with acetyl coenzyme A form HMG coenzyme A with the help of HMG coenzyme A synthase. That was the rate limiting step for the ketone bodies. But here HMG coenzyme A will combine form our mevalonate with the help of enzyme HMG coenzyme A reductase. So this HMG coenzyme A reductase will be the rate limiting step for the cholesterol. But while in the case of ketone bodies, the rate limiting step was HMG coenzyme A synthase. Okay, so basically this HMG coenzyme A will form our ketone bodies, it will form our cholesterol, and it will form the leucine compounds. Okay, so next is the bile acids. Next is our bile acids. All of us know that we have two type of bile acids, that is primary bile acids and secondary bile acids. Both of them are 2 in number. Primary bile acids are produced in liver and secondary bile acids are produced in intestine. The primary bile acids are, it can either be cholic acid and it can either be chenodeoxycholic acid, while secondary bile acids are deoxycholic acid and lithocholic acid. First of all, you should remember the primary and the secondary type of bile acids with their examples. Okay, so what are the derivatives of bile acids? Basically, it is the steroid hormone, vitamin D. Derivatives of cholesterol are basically the bile acids, the vitamin D, and that are the steroid hormones. So this was a short video about the ketone bodies, the cholesterol, and that are the bile acids. Thank you for watching.